Good morning, good afternoon, or good evening, depending where you are, and welcome to the Global Health Institute next installment of our Global Health Tuesday webinar series entitled Bird Flu in Cattle. Thank you for joining us. My name is Jorge Osorio, and I'm the director of the Global Health Institute. Our monthly Global Health Tuesday webinar series features researchers and practitioners from the University of Wisconsin-Madison and around the world, and highlights the complexity of global health challenges and the many kinds of expertise needed to address them. And if you have been attending our webinar series, you know our foundation, UW-Madison, begins with the Wisconsin idea, a practice that takes knowledge and research out of the classroom and into the real-world settings. Today's sessions continues with that focus. As many of you know and are aware of or have read in the news, the H5N1 avian influenza, or more commonly known as the bird flu, has been affecting our U.S. dairy herds. It has been detected in milk, and a couple of dozen human cases have also been reported in the U.S. What does this mean when a virus that historically targeted birds and is now being found in mammals? And what steps are being taken to learn more about the current virus and potential mutations that mitigate a potential pandemic? Today's discussion will explore these questions and more of other issues from our STEAM panel. Before I introduce our panelists, a couple of quick housekeeping items. We are recording today's session, and the archive video will be available at our website at ghi.wisc.edu. And second, we are monitoring the chat, so please share your questions and comments for the panelists there. So with that, let's get started. Today, we begin by welcoming Amie Eisfeld. Amie is a scientist in Jorge Kawaoka's lab in the Department of Pathobiological Sciences at the School of Veterinary Medicine at UW-Madison. She is a molecular biologist whose research focuses on RNA viruses that have caused major epidemics or pandemics in humans, including influenza and Ebola viruses. Her principal interests are in understanding viral and host factors that regulate viral pathogenesis. Amie is also a member of GHI's advisory committee. Welcome, and thank you for joining us, Amie. We're also welcoming Dr. Peter Halfmann. Peter is an assistant professor at the Influenza Research Institute within the Department of Pathobiological Sciences at the School of Veterinary Medicine at UW-Madison. His research is dedicated to understanding emerging and re-emerging viruses, including filoviruses, coronaviruses, and influenza viruses. Peter is also a member of GHI's advisory committee. Welcome, Peter, and thank you for joining us. Next, we welcome Dr. Kit Poulsen. Kit is a specialist in large animal internal medicine and has been a diplomat of the American College of Veterinary Internal Medicine since 2008. He holds an appointment in medical science at the School of Veterinary Medicine and is the director of the Wisconsin Veterinary Diagnostic Laboratory at UW-Madison. Kit is also the president of the American Association of Veterinary Laboratory Diagnosticians and has active service projects in the United States, South America, Asia, and Africa, which focus on infectious and zoonotic diseases and sport market development. Welcome, and thank you for joining us, Kit. And finally, we also like to welcome Nailah Smith. Nailah is a veterinary medical officer in USDA Animal and Plant Health Inspection Services Office of Interagency Coordination. Within this office, she's a member of the One Health Consortium unit and supports collaboration with federal One Health partners such as the Centers for Disease Control and Prevention and the FDA. Prior to joining the USDA, she earned her dual DV MPH at the University of Wisconsin-Madison. And we'd like to collaborate with a Bayer alumni. And so let's begin our discussion with you, Nailah. Can you please share with us your thoughts about the bird flu in cattle and what the USDA thinks about this? Thank you.
So much, Dr. Osorio. Let me pull up my screen. So I'm going to give a pretty high-level overview of sort of the HPAI in dairy cattle event. So, just to start off with running our numbers, as we all know, on March 25th, 2024, USDA National Veterinary Services Laboratory confirmed the first detection of HPAI H5N1 clade 2.3.4.4b genotype B3.13 in a Texas dairy herd. Phylogenetic analysis and epidemiology support a single introduction into this novel host, followed by onward transmission. So as of October 29th, 2024, the total confirmed detections for this incident includes 388 premises in 14 states. So in the last 30 days, there have been 145 confirmed cases in 2 states, and we're really seeing the vast majority of our cases coming from California. I want to talk a little bit about APHIS's action strategy. So once we got over the initial shock of seeing such an unexpected disease and such an unexpected host, got to work on developing a plan to mitigate virus spread and hopefully ultimately eliminate this virus from the national dairy herd. So we really want to do 4 things with our strategy, and that's understanding virus in cattle, so pathogenesis and transmission, understand the distribution of the virus, mitigate movement of the virus, and provide support to producers. I'm going to touch on some of the activities that we've been doing in each of these spaces. And one of the first things I'll touch on is our federal order. So on April 24th, 2024, APHIS announced a federal order mandating testing prior to the interstate movement of lactating dairy cattle and reporting of positive influenza A test results in livestock. So the mandated testing as part of the federal order includes testing of suspect or animals in contact with suspect animals, and then testing for any associated species. And this is all offered at no charge to the producers. So clinically affected or lactating dairy cattle from herds that have tested positive for influenza A were not eligible for interstate movement for 30 days from the most recent collection of any sample that tests positive from any individual animal in the herd. And then after the 30-day period, the animals must be tested again. So that was one of the first tools, in addition to kind of the laboratory testing at the National Veterinary Services Lab and our National Animal Health Laboratory Network, to really start to, get this action strategy rolling. We've done a lot of field activities since, March of this year. We've worked on administering really intensive epidemiology questionnaires. They're voluntary. We'd like to get them to as many producers as we can. About 56% of producers volunteered to complete interviews. There is on-farm field research going on in different states, and there is wildlife and peri-domestic species sampling. Our wildlife services team has been really amazing and made themselves available whenever we get a new dairy premises with B3.13, or even if there are poultry premises that are associated with B3.13, they are able to collect samples from peri-domestic species within and around those premises that are affected. And then we also send epidemiology support or strike teams to states that request it. And this is really to support a more robust field epidemiology investigation and to support rapid data collection and analysis. And I'll just pause here to point out that each of the circles next to those topics, you can see what states that they've been applied to. So it's pretty much throughout every state that has been affected to date. With all of that field activity comes a lot of data analysis. And so, those epidemiology questionnaires, I believe they're 22 pages. So it's a lot of data to work through. We've already had a couple of iterations of an epidemiological brief. So this is summarizing on a national scale the information that's coming out of those questionnaires. And those are up on our HPAI and livestock website. There's also on-farm sampling, recruitment and coordination, and summary of those results, and then production movement and data analysis. So just a lot of moving pieces with our field activities, and keeping as much up to date on our website as we can as those get completed. So another tool that is part of this strategy is our Dairy Herd Status Program. So on May 30th, 2024, APHIS announced the implementation of an H5N1 Dairy Herd Status Program. So this program is a cooperative effort between APHIS, regulatory state animal health agencies, the dairy industry and its stakeholders, and dairy producers. And this provides support for ongoing H5N1 testing to detect, respond, and better understand this virus and reduce the risk of further spread. So it has the benefit of offering dairy producers more options to monitor the health of their herds and conduct required pre-movement testing on a herd level. And this facilitates moving cattle interstate under the federal order. So far, 64 herds have signed up for the program or a state equivalent program. I'll talk a little bit about some of the financial support that we're offering to producers as well. So this is to assist with biosecurity planning, reimbursement for any sample collections of testing, offsetting costs, and then also funding for sick cow milk disposal, reimbursement for veterinary costs and fees. So this is really to try to cover those dairies that have not yet detected H5N1, trying to help them with the tools they need to prevent this in their herds or catch it early. And then those dairies that are already affected, really offsetting some of those costs that come with that. And I'll note that there's another financial program that is run by another agency, FSA. It's called the ELAP program. And so there are even more financial options for affected dairies through that program. So some of the key messages that we're sharing is that the spread of H5N1 between states is linked to cattle movements. So this is a little different than the poultry outbreak, where we see in our poultry facilities that it's a lot of independent wild bird introductions. What we're seeing in dairy is that it's really linked to cattle movements with further local spread between dairy farms in some states. And disease spread between dairy cattle farms is likely multifactorial. So there are some direct and indirect transmission routes. And we're really seeing that biosecurity is key to mitigate the risk of disease spread. So the next steps for APHIS really focus on encouraging the dairy industry to participate in some of these newly developed programs to sort of clean up herds and avoid spreading the infection, and ultimately wish to eradicate the virus from the national dairy herd. So thank you.
Great. Nailah, thank you so much for such a wonderful presentation. And now let's hear from Keith to share information from the Wisconsin Veterinary Diagnostic Lab. Keith, please share with us your thoughts on your slides.
Forgot to unmute there. Hopefully people can see my slides. I think I kind of had some thoughts on my drive. I'm up at Food and Farm in Plover to talk to some dairy managers. And, there's one thing I really want to make sure is I'm not a virologist, I'm a veterinarian, although no self-respecting veterinarian would wear a white polo shirt in a dairy barn, but it made a great picture. And I'm gonna switch gears, I think a little bit, and talk about the context of what's happening. And because I really think that's what the Global Health Institute is about the context of wherever we are working or what we're doing, or research or outreach or service projects. So I'm going to skip this slide. Actually, we know a lot about testing cattle, and I think Dr. Smith talked more about the surveillance programs and ELAP that require surveillance. We know that bulk tank sampling and either or PCR or ANTI is effective. And this is the National Animal Health Laboratory Network that has plenty of capacity to do that. The rate limiting factor has nothing to do with the labs and our capacity and awesome technology. It has everything to do with this milk sample vial right here. This is a standard milk vial. It's a flip top 2 ounce. They typically are overfilled, and every time you pop that top, it spreads milk and virus everywhere. So we need to think about that, and it's all about logistics coming from the farm to the lab. It's not about the lab. So, but I'm going to keep moving because just for time's sake. And I showed this data before, and I'm going to show you one other piece of data. The most important part of this is a longitudinal study of a herd that was collecting milk samples before and after outbreak. And because their neighbors were being affected in Idaho, this data is from Iowa State. The most important part of this data, there's 2 different things here. One is that we know that dairy cows are shedding virus in milk, and we can find it in the bulk tank with pretty high reliability, 100% at a CT of 32 or less. In a tank that represents 1,000 animals, which is about the size of those direct ship tanks on the roads. We can find them 14 to 16 days before they break with clinical signs. That's really important, and the reason why surveillance is so key. Not necessarily to completely eradicate virus to begin with, but at least we know where virus is, where it isn't, and we can prepare farms when virus is in the neighborhood. The second most important part here is that these bulk tanks have a pretty strong antibody response. Cows do seroconvert, they produce antibodies, not nearly to the level that poultry does, but they do seroconvert. We think that seroconversion or the exposure immunity right now, Iowa State's preliminary data is about 6 months. We are hoping for longer, but that shouldn't really surprise anybody. But here the key thing is really looking at where this intersection is at right about a week after start of clinical signs on farm. And dang it. And that's a question that we have is that if there's antibody in the bulk tank, is there virus in the bulk tank? This is pretty preliminary data, and you're going to hear some more data from the Influenza Research Institute with Dr. Halfmann and Amie. Dr. Eisfeld. But we know that looking at 16 days after, this is just limited data, that 16 days after we're getting CT values in the 30s, so it's positive. And we also have strong ELISA positive. This is a different herd in Idaho, but we also found no culturable virus in there. This isn't unique, but we haven't been able to collect these to get this data published. But this is really key for both public health and understanding how long we need to quarantine farms to maintain business continuity. So the most important parts of the things that I want to talk about actually start here with biosecurity and then move into where we need to go. So this is data traceback, and we got this from Maggie Baldwin, because the only state in the country that is doing regular surveillance right now is Colorado, where they're on 1 week, and their last tests in September were all negative. Massachusetts is also testing all dairies once a month. They are all negative, so the 2 farms, but Massachusetts has never been affected. So Colorado is the only state that has been affected that is testing all dairies. California is moving to that component. The big difference between Colorado and California is that there's about 100 dairies in California, or in Colorado, I'm sorry, and 85 of them are in 2 counties. They're 80% positive at this point. And California has, and that's 250,000 cows. California has about 1.7 million cows, and they have 1,100 dairies. Now, just comparing that back to Wisconsin, we have 5,400 dairies with about anywhere from 1.2 to 1.6, depending on the year, million dairy cows. So obviously our herd size is quite a bit smaller, but here's the trace back. The green dots are farms, the yellow, maroon reddish dots are service providers, whether that's electricians, people that work in the parlor. You can see how interconnected all of these premises are. It's really important to point out these 2 poultry farms. This farm right here has been depopulated 3 times this year. These 2 poultry farms represent 8 million birds. So there is a lot of connections. And even with these poultry facilities, which are shower in, shower out, it's exceedingly difficult to completely use biosecurity to help. Biosecurity is our most important component of this disease at this point, mainly because we have no idea where virus is and where virus isn't. We have a general gist, but we really don't know across the country, and it's a national industry. So I think this really highlights the importance, yet how complicated it is. Dairy is light years behind poultry and swine for biosecurity because we've never had to deal with this in the past. So where does this go? I want to spend the most amount of time here. I had a very interesting conversation with a veterinarian in Kansas on the way up to Plover today. And I asked, because right now we're pushing pretty hard for better surveillance. USDA has come in light years in the last few months. The ELAP funding and the herd status program is like nothing we've ever had before. And the type and the amount of work that had taken is really impressive. Unfortunately, it's not moving the needle. We know that only 70 herds are in the ELAP program when there's over 350 that have identified, and the estimates are that the total number of herds, that's only about 10% of total number of herds affected. We also know that the status program is under 50 because it's too expensive, and it really there are too many other complicating factors that are happening, and many farms aren't signing up because of adjusted gross income limitations. So it can be better, and I think that's where the support of industry really needs to be leveraged, because the consensus for the industries of pork, swine, dairy, and beef nationally is that we need to do better in order to protect all of our food producing animal industries. Now, I was talking to Tara, back to Tara. I said, Tara, if we're thinking, this is Dr. Barnhart, she runs some very large dairies in 8 different states. She lives in Kansas, Southwest Kansas. I said, what's the palatability of running more intensive surveillance in large dairy states in this Texas panhandle after you've been there? You're going on about a year. This started about a year ago. They really started noticing it as a problem in December and January. And she said, actually, that's quite high. She said, but the most important part about surveillance is the context that goes around it. And I think that's the key component of why this is so important for the Global Health Institute. The context here is a year ago, milk price was in the toilet. It was really low. It was less than what it costs to make it. So farms were using other sources of revenue in order to stay afloat. Feed prices were also exceedingly high. So what does that mean? That means that we weren't pushing cows very hard at that point to make a lot of milk because it just didn't have that return on investment. And then what happened is H5N1 came in. We don't really know how, and we don't really know how this is moving. We think it could be with movement of lactating animals, but we can't prove it. That's what it logically it would be, but we still really can't prove it. And I think the Influenza Research Institute is going to tell us more about this. I'm about to get inundated with kids here. But now it's all very different. Milk production in the Kansas and Texas area is very high, very high. Systematic cell count is very low. Feed price is very low. So there's a potential for substantial revenues coming in the next year. Those dairy owners in Kansas and Texas are very anxious because they have no idea where virus is, where it isn't. And if virus starts to move again. So I think they would be much more applicable to surveillance, and they know that they're a part of the problem in the spring. And I think they're willing to move on. So looking at the context of what the people on the ground are doing, we know National Milk Producers Foundation our federation had a meeting in September. AVMA had a roundtable end of September. National Academies of Science, Engineering, and Math had a meeting last week. We're having a meeting right now with poultry and egg producers. They're all saying the same thing. Decouple this as an animal disease from milk as a food product because pasteurization is effective. That's a key component. This is an animal health disease, and the animal health disease has to be managed by the subject matter experts that know how to do this. They all agree, the consensus is that surveillance is absolutely critical, but it has to be consistent across state lines because it's not an industry of just Wisconsin. It's not an industry of just Texas or California. It's a national industry. Animals are moving all the time. In order to do that, we have to be able to protect that data, just like we do with testing records for other things like Johne's disease. And that's a blast from the past. I thought I saw Dr. Patton on. She managed that. We have to be able to define quarantine. We never put animals in quarantine without understanding how to get them out. And that's very unclear, and every state is doing something different, creates problems. We have to guarantee market access, because if you can't sell your product, that's a big problem for the context of those dairies within our communities and our states. We talked about antibody testing. That certainly should be used in our strategies, and we can do more about that. And we have to define what elimination looks like for business continuity and animal welfare. We can't just stop. It's not a widget factory that you can just turn off the button. That won't work. So I'm going to stop here because I do want to leave time for questions. And I know there's a lot of great information coming out of IRI. So thanks.
Well, thank you, Kit. Fantastic information. Thank you so much for your presentation. And I just want to remind the audience to please submit your questions in the chat. And so we can then later on go to our Q&A session. So now let's hear from both Peter and Amie about the research that is happening at UW Madison in avian influenza and bird flu in cattle. Thank you.
So Amie and I are going to round out this conversation by talking about the research aspect of bovine H5N1. We're out here at the Influenza Research Institute, which is part of the School of Veterinary Medicine. So what we want to do is give a brief summary of high-path avian influenza and to try to cover how did this originally start back in 1996. How did we get to the fact that now the virus is infecting cows? What were the origins, the spread of H5N1 in mammals, and why that is such a big concern? And then Amie will go into our research and characterizing some of these H5N1 viruses that we've obtained from both cows and human cases. So again, this started back in 1996 when the H5N1 Guangdong lineage emerged in geese. And this resulted in a first infection of humans. 18 humans were infected in Hong Kong in 1997, and 6 died. The virus then continued to spread in Southeast and Southwest Asia from 2004 to 2006. And then we saw a massive explosion of the virus circulating in avian species throughout Asia and going into Europe. So to date, 896 human cases have been clinically confirmed. I think that's a number you should remember as we talk about what's going on here in the United States. Out of those 896 human cases, 463 were fatal. So that's roughly a 50% case fatality rate. Again, a number to remember as we continue to discuss this virus. There's little evidence of human-to-human transmission, which is great. However, work from our group and a group in the Netherlands have shown that the virus can mutate. Influenza virus is a great virus to have mutations occur within its proteins. And we've shown that mutations in the particular proteins of the virus can enable transmission so that the virus can transmit from mammals to mammals. And then until recently, high-path H5N1 influenza virus was largely a problem that was confined to the Eastern Hemisphere. But then roughly in 2020, there was a reassortant, another aspect of influenza virus that they're prolific to reassort with other influenza viruses. And this now has caused or created the novel clade of 2.3.4.4b genotype, which is the virus that is circulating in the Americas through the transatlantic flyway, allowing this virus now to basically be detected in every country in the United States. Those are the countries shown in orange. The countries that are shown in white probably do have H5N1 circulating, but the surveillance is just not there to detect it. So our concern as researchers about avian influenza virus is the fact that avian influenza virus is an avian virus. But again, it's the ability of avian influenza virus to mutate and eventually acquire or potentially acquire amino acids that's going to make the virus more mammalian-like. If the virus becomes more mammalian-like, then we have a higher chance for this virus to replicate efficiently in humans, other mammals, and then the potential of the virus to transmit in humans. The first indication that this virus was more ready to transmit from mammal to mammals was in late of 2022 in Spain. This farm, or this mink farm, realized that they had an increase in mortality in their mink farm. They assumed this was from COVID or SARS-CoV-2. Turned out that it was actually the clade 2.3.44b virus, and they had to call all 50,000 mink on this farm. But this was the first example that the virus, first compelling example that the virus had been able to transmit from mammal to mammal. As the virus circulated in the Americas then, you could see it go down the coast of South Africa or South America. And we saw the transmission of the virus from birds to wild mammals, marine mammals. This included initially 600 sea lions. Later to find out that the virus was actually had killed over or close to 4,000 sea lions in Peru. The virus continued to go down the coast and affect the sea population or seal population in Argentina. This was resulting in over 17,000 elephant seals that died in Argentina, and again showing that the virus had the ability to go from birds to mammals and then transmit between mammals. And as Dr. Smith alluded to, this was a surprise then in January of this year that farmers were talking about a reduction in milk production in dairy cows. A few months later, it was confirmed that bird flu had spread to dairy cows and was affecting the states of Texas, Kansas, and possibly New Mexico. Dr. Smith showed this picture showing that the introduction of H5N1 avian influenza probably started as a one event from birds into cattle, starting in Texas, and then the transport of cows throughout the United States had spread the virus, showing that 380 herds had been affected in 14 states. And it was all over the news that the virus genome, not infectious virus itself, was being detected in cow's milk and in tissue of these cows. And then to date, for the human cases in the United States, we've had a total of 34 humans infected with cow H5N1. The striking difference here is that unlike in Southeast Asia, what we're having in the United States are more mild respiratory symptoms, a lot of eye infection, no deaths to report, and human-to-human transmission doesn't seem to be clearly observed, with the potential exception with the Missouri case where a person in Missouri was infected. They didn't have any known contact with poultry or cattle, and there's potential that person had a close contact with the person that lived with that was potentially infected by serology.
Okay, so I'm gonna start talking now. So the first thing that I wanted to talk about is just a little bit is bovine H5N1 in cow's milk. So as Pete already mentioned, earlier reports indicated that viral fragments or the genetic material but not infectious virus were present in commercially available milk. So people went out, bought milk off the shelves, and they were able to use PCR to identify the presence of influenza there. And very early in this epidemic, we received some milk samples from collaborators in Texas, and we used that naturally infected milk with our under laboratory conditions. We were able to show that heat could substantially reduce the amount of virus that was present in those samples. And then furthermore, folks at the FDA also used commercial pasteurizers to show that there was a complete inactivation of virus. So mainly the take home that I just wanted to briefly mention here is that consuming commercial milk that's pasteurized is safe, but drinking raw milk that has not been pasteurized is not necessarily a good idea in this particular context. But for the rest of the talk today, I'm going to switch gears and talk about work that we're doing in animal models. So the basic characteristics of bovine H5N1 viruses are unknown. So there's been a lot of work in avian viruses from Southeast Asia that we've done in the laboratory over the years, but we didn't really know, if that equates to, or if the characteristics are similar to what we might observe with bovine H5N1. So the goal of our studies that we've been performing over the last couple of months are to define the potential risks of bovine H5N1 to humans by examining its effects in well-established animal models of influenza infection. So the work that I'm going to talk about today covers pathogenicity and transmission in the most commonly used animal models of influenza infection, which are the laboratory mouse and ferrets. And the data from these studies can be found in the two publications that are listed at the bottom of the slide. So I'm going to briefly touch on pathogenicity studies. We don't have a lot of time today, so I'm just going to summarize these, and I'm not going to show any of the data. So in the mouse model, we found that bovine H5N1 induces severe disease after either oral ingestion or respiratory infection. The LD50 of these animals was quite low, about 30 platforming units. What that means is that you need only about 30 infectious viruses to kill 50% of the mice that are infected. We also found that infection by either route can lead to systemic spread. And what that means is we found virus in the respiratory tissues and in non-respiratory tissues like blood, liver, kidney, and spleen. But then we also found virus in the eyes, mammary glands, and muscles, which is relevant to what we're observing in both cattle and humans in the context of the bovine outbreak. So people had not looked at mammary glands before, but we found pretty consistently that the virus can spread to and replicate in the mammary glands. And in the ferret model, as in mice, we observed that respiratory infection could result in systemic infection with wider tissue tropism than had been previously appreciated. So I'm going to focus a little bit more time on transmission because this is something that is really important in the context of the potential for causing the next human epidemic or next human pandemic. So the typical route of influenza spread in humans is by respiratory droplets. So whenever a new virus like this emerges, one of the most important questions that we want to ask is, can the virus spread by respiratory droplets? And so to do this, we carry out studies in the ferret model, and this is the approach that we use here. So just to briefly go over this, we infect animals, donor animals, with bovine H5N1 or our control seasonal influenza virus at 10 to the 6 plaque-forming units or 10 to the 6 infectious viruses. And then one day later, we add contact animals in an adjacent cage that's separated by about 5 centimeters. What this means is that the two animals do not have any direct contact with each other, but aerosols are able to transport between the two different cages, and so virus spread can occur through the aerosol route. After these animals are placed in cages together, we take a nasal swab from those animals to test the virus titer every other day. And then for any animals that survive the infection or the experiment, we also look for hemagglutination inhibition titers, or basically antibodies in the blood, to see if there's any potential exposures that we can observe in that way. So the data from this experiment is shown here in this slide. So these are nasal swab titers. On the x-axis, I'm showing time, and on the y-axis, I'm showing the amount of virus. On the left are the donors, and on the right are the contacts. And what you can see on the left is our donor animals, both the bovine H5N1 infected animals and those that were infected with the Asumi H1N1 seasonal influenza virus, exhibited substantial virus replication in the nasal tissues, which is what we would expect. Then when you look at the right-hand side, we're looking at the contact animals. So these are the animals where respiratory droplet transmission occurred. And if we're looking at the bottom panel, we can see very efficient respiratory droplet transmission with our seasonal influenza virus, which is what we expected. This is the control for the experiment. But there was no virus detected in the nasal tissues of the bovine H5N1 infected contact animals. However, one contact from the bovine H5N1 experiment did seroconvert. That's indicated by the red circle here. So that means this animal had antibodies against influenza in its blood. This suggested that aerosol transmission occurred, but that it was quite inefficient. Okay, so while this work was under review, the CDC reported 33% aerosol droplet transmission of a human isolate of the bovine H5N1. So here I'm showing a phylogenetic tree of different bovine H5N1 viruses, which are shown in the blue, as well as the purple, indicated by the purple arrow, a human isolate that was collected from the eye of an infected human in Texas very early during the outbreak. So I will just briefly note that this human isolate is different. You can see that by the different branches on the phylogenetic tree from the majority of, or basically all of the bovine H5N1 viruses that have been sequenced to date. There's a table at the bottom of this slide that shows the amino acid differences between our bovine H5N1 virus and this human isolate. And the most important amino acid difference that I will point out here is that of PB2-627. So this is a K or a lysine in the human isolate. This particular amino acid mutation is mammalian adapting and allows for greater growth or higher growth of the virus in mammalian tissues. So this was potentially of concern. And so we wanted to look at the ability of this virus to grow and spread by the aerosol route in ferrets. And so this data on this slide summarizes our aerosol transmission studies with this human isolate. In this experiment, we infected animals with different dosages, 10 to the 6, 10 to the 3rd, 10 to the 2nd, or 10 to the 1 infectious viruses. And we observed transmission in 17 to 33% of animals in all cases. Another important thing that I need to point out here is that all of our donor animals, no matter what dosage they received, died within 6 days post-infection. This is very unusual for any influenza virus in the context of a ferret model, something that we've not observed with other viruses in the past. So the human bovine H5N1 isolate is extremely pathogenic and consistently transmits in the ferret model. So overall, our conclusion, I know I went through just a little bit of data and quite fast, and I'll just mention again, if you want to read more about this, you can see it in our recent publications. But bovine H5N1 viruses are highly or extremely pathogenic in mammals, and our data show that they do transmit by the respiratory route in ferrets, though it's not efficient transmission as of right now. We believe that every effort needs to be made to contain these H5N1 outbreaks in dairy cattle in order to limit the possibility of future human infections. So I've got one more slide to briefly touch on today. There are some unanswered questions that we're interested in addressing in our future work. So bovine H5N1 is highly pathogenic in animal models of infection. And also, as Pete already talked about, we see high case fatality rates in human infections with avian H5N1 influenza viruses in Southeast Asia. So one question that we have is, why hasn't severe disease been reported in any of the human cases in the United States? We do have some ideas about this. We're all exposed to influenza viruses year over year. Our pre-existing immunity may be partially protecting us from severe infections. There also may be a route effect. So if people are primarily infecting themselves by maybe rubbing their eyes, maybe they're not getting a lot of virus into a tissue that doesn't really support extremely high replication, and maybe there's not access to the body in the same way that you would get through a respiratory infection. And then the other possibility is that active surveillance here in the US is catching mild cases that might not have been observed in Southeast Asia, where active surveillance is not taking place. Another question that we have is, what mechanisms are driving pathogenicity and transmission in animal models? And we have a lot of work that's going on right now in the laboratory in order to address those questions. So that's all I've got to talk about today. And yeah, so I guess we can give it back to Jorge.
Thank you, Amie. Thank you all for such a wonderful presentations. And again, to folks in the webinar, we open for questions. So please, if you can write your questions on the chat, that would be great. We had a question about what can farmers do if they detect virus in their vicinity. And maybe, Keith, can you talk about that, but also comment about other species in the farm that can be susceptible to avian influenza from cattle? Can other species be susceptible as well?
Sure. So on many of our dairies, what happens is you start seeing affected neighbors, and then you see that, and then all of a sudden, sorry about the you'll start seeing cows that go off feed. You may also see, we've heard anecdotal reports of birds dying on farms, although that's really difficult to understand or have good data on. We've also seen spillover to cats and then other rodents like house mice or barn mice. So the best thing to do is, there's people on the call, is contact your veterinarian and contact the state animal health department or DACCAP. So Dr. Conkle, Dr. Roney, and Dr. Patton, I think, are on. I think Dr. Belay is too. That's their job. They respond to that, and then they work with the veterinarian in the diagnostic lab to help understand what's going on. We also have regular surveillance, like what Dr. Smith was talking about, for animals that are moving or going to exhibition. So hopefully that answers your question.
Yes, thank you, Peter. And Dr. Smith, thinking about the state of Wisconsin, it's nice to know that the state has been remained free of cow bird flu. Can you comment on, your thoughts on that, being such a major dairy state and some other ones as well? Is our surveillance programs quite efficient?
I think Dr. Poulsen could probably speak to this better than I can, but I'll take a stab at it, then I'll see what he thinks. So what I can say is that I know that Wisconsin is engaging in our dairy herd status program, and they are testing at the National Animal Health Laboratory Network there. And so they are engaging in the surveillance activities. I anticipate that they would be keeping a very keen eye on their herds for any signs of illness. And so, looking for, the virus there. One thing that Dr. Poulsen touched on, and I think that we're kind of seeing, with the data that we have available, is that some of the herds in Wisconsin tend to be a smaller size. And the states with a lot of affected herds, they're having much larger herd sizes. So we don't know exactly why we're seeing this trend or kind of what's behind it, but I think that might be something to consider. But I'll let Dr. Poulsen add any thoughts on that.
I think there's a couple of big changes, and Dr. Smith's right. We have 4 herds that are doing monitoring. They're all managed by UW, and I don't know that they're officially part of the status program yet, or hopefully they are, to utilize those resources. But we don't import large numbers of lactating animals. We just don't. Michigan does. It's part of the Dendalk and Fairlife model, and they move large 30 to 50,000 lactating animals per week as part of their business model. So that's a huge impact. So because we don't have those very large farms in the area, I think that's one big thing that's protecting the state. Although it wouldn't surprise me if we came up with a positive tomorrow. The thing that makes me think I'm, with high relative confidence that we don't have virus in the state is that there's a lot of labs that are looking at commercial product as kind of I don't want to say crude, but another way to do surveillance. That's how Canada is doing national surveillance right now, is looking at commercial product. So that is one way that we can think about that. And if we were to see a positive there, we do see positives in products, but when we break them down into individual components, it's from the dried milk protein, and guess what? It's sourced from the states, right? So I do think I commend the folks that are doing that research. But the difference in Michigan definitely is they are part of that, and then they had spillover because they have people whether they're communal living or they're working on multiple farms. They composted a bunch of dead birds right next to a 200 cow dairy. That was a direct link. So there's a lot that happens here, and a lot that we don't know of because we don't have cohesive national surveillance. We just don't. And if we don't know where virus is, and more importantly, where virus isn't, it's hard to then describe what's protecting those dairies. So there's a lot that needs to be answered yet. And I know everyone from USDA, DACAP, and industry working very hard to figure out how we can make this happen.
Thank you, Keith. That's a great answer. Thank you so much. There are a couple of questions that I think are really interesting, and it's about how this virus could infect other animals, especially wildlife, and also if the cows shed virus in their manure and we're spraying that in the field. So how does the virus could survive in the environment? So 2 questions. Can wildlife like deer and other ungulates be infected? And second, how does the virus survive in the fields? Amie, would you like?
Yeah, so I could maybe say a little bit about this. So there have been quite a few detections of these clade 2.3.4.4b viruses, which is what the bovine H5N1 viruses are, in other animals like skunks, foxes, mountain lions, bears, other kinds of predatory animals, in addition to lots of avian species that are in the US. So whether or not the virus can transmit between those animals is not known. There are differences in all of the animals, and a virus may not be adapted to a particular type of virus or a particular type of animal. Maybe it doesn't grow extremely well in some, maybe it does, maybe it spreads in some, maybe it doesn't. We don't know all of the details of that, but certainly there is a possibility that there could be mammal to mammal spread in the wild. So whether or not that puts us at risk in the future, certainly it's a possibility. If the animals that are being infected, like foxes, are pretty, solitary and not, in contact with a lot of other animals, then maybe we don't see a lot of spread. But as Pete mentioned at the beginning, there's been a lot of spread in sea mammals, and then a lot of spillover and spill back between sea mammals and birds. So I kind of think this is not going away for a while, and this is going to be something maybe this is something that is going to evolve and change with time. One of the main things that we can say is that the more replication that we do see in mammals and in humans, the more likely we are to develop some kind of a mutant that would be of higher concern for humans.
Thank you, Amie. And you think that virus will survive in nature, in the environment for a long time?
So that I don't know for sure. We have done some studies under laboratory conditions and just virus in milk. So this is not necessarily, virus in water, but virus in milk, at least at 4 degrees, is stable for several months. Whether there may be some data out there about environmental stability. I don't have a good handle on that, so I don't want to talk about it too much. Whether it can survive in manure, that might be a question for somebody else. I'm certainly not an expert, but I do believe that people are doing some wastewater surveillance for these kinds of things too. So maybe somebody else could address that.
Yeah, thank you. Thank you, Amie. Peter, there's a question about this virus being a virus that can do reassortment, that can make reassortments, and if there is a human being, infected with this H5N1, but also might have a regular seasonal flu. So is there a chance here that this virus could reassort itself and become more pathogenic?
Yeah, so I think that's one reason that California has ordered 10,000 to 15,000 more seasonal influenza vaccines for this year, because there is a worry with these farm workers that are not probably getting adequate PPE to use on these farms, that they become infected with H1N1 or H3N2, a seasonal human influenza virus, and then potentially get infected with an H5N1, where we could have a reassortant virus that would come out that could be more transmissible or more pathogenic in humans. On the other side, we're also still worried about the H5N1 virus replicating in mammalian species in a acquiring mutations that make it better for humans too. So there's kind of two sides that we're looking at with the increase in pathogenicity and transmission of H5N1.
Peter, could you
can I
talk
ask-
about the importance of pigs there?
Well, and then
Sorry,
pigs.
Jorge.
Yeah,
So
please.
pigs can be infected with both avian and human influenza virus. They have receptors for both virus. So pigs are the big mixing vessel. So if you don't have a human that could serve as the catalyst of a reassortant virus, you have pigs, which are also co-hosts on potentially on farms that have cattle, that have chickens. So you could have a reassortant event in pigs too. So that's another concern. But I think USDA is hopefully pushing surveillance in pigs as we come into the fall season here.
Any comment they can make on that, Nailah, on surveillance in the swine population?
Yes. So I had to pull up my notes because I am not on the swine team. So to be sure I have the correct information. Wildlife Services actually has been conducting serological surveillance for influenza A virus in feral swine since October of 2023. So they've also initiated some limited enhanced targeted surveillance in 3 states, so California, Texas, and North Carolina, testing nasal swabs by PCR. So, you know, that has been kind of our start at enhanced surveillance in the feral swine population. And then, as you know, the swine industry has some really incredible biosecurity tools under their belt. So we have our national swine influenza surveillance with that industry as well.
Great, thank you. And then can I ask a question if other countries near the US are doing cattle surveillance? You know, if Mexico, for example, that has very strong dairy industry, Canada and some other countries in the region are doing cattle surveillance. Can someone comment on that?
I'm
Keith,
not
would you know?
Oh,
Would you,
I was
Nailah,
going to say,
go ahead.
Nailah, did you want to start?
I can start. I kind of know what you had mentioned, that Canada has been doing that surveillance in their pasteurized milk products. And then I know we've been in close communication with Mexico as well. We presented on sort of a binational call with them about the dairy flu situation. But so far, I don't think that they have had like a national surveillance system at this point, but I imagine that they're just keeping an eye on any potential, introduction.
Keith,
I
can you
think
comment?
what complicates Mexico too is there's other endemic H5 and H7 strains in the avian population, right? So, and they use a lot of vaccine. So, and that's really kind of the question in multiple different areas, or in the EU as well. We get quite a few questions. Colombia stopped the live animal and germplasm import markets because of that, but then they reopened. Turkey has also changed to no live animal imports. And I think in general, it's really complicated when you think about viruses, where it isn't, who's using vaccine, and then you put in a bunch of political questions with trade issues. So it makes it quite complicated. But other than the United States at this point, I'm not aware of any active surveillance of either bulk tanks or.
Thank you. Thank you, Pete. So great. So I think with this, we pretty finished with our questions, and I'd like to ask you to join me to thank our panelists, Nailah Smith, Keith Poulsen, Amie Eisfeld, and Peter Halfmann, for sharing all their knowledge and their perspectives on the bird flu in cattle. And I also like to thank you all for joining our webinars, and also for engaging in discussions and comments. And finally, I'd like to invite you to our next Global Health Tuesday webinar, which will be on November 19, and it will be about preparing for future pandemics, and it will be the Out Pandemic Defense Coalition Network. Please make sure that you visit our website at ghi.wisc.edu to learn more about the activities that we're doing at GHI, and also about the video from this webinar. And I hope everyone has a wonderful day, and I'll see you next time. Thank you so much.
You.
Thank you.